This re- this recording is over Chapter 10. We're going to talk about DNA structure and composition. Now, it wasn't too long ago that there was a big debate amongst molecular biologists as to what material inside the cell actually housed this heritable material that was being passed on from parent to offspring. So we talked a couple lectures ago about the chromosomal theory of inheritance, but before we fully understood what DNA was, even though we could see some of the chromosomes under a microscope, we weren't able to ascertain what is that material. And so there was a big debate about, are proteins responsible for that genetic material, or was DNA? Now, we know that chromosomes, they're made up of both proteins and nucleic acids. So even when we isolated out a chromosome and we broke it apart, we found both proteins, nucleic acids in there. And the reason why this debate was hotly contested was because in humans, we have around 20,000 different proteins, as opposed to only 20 different amino acids that can be found, that make up those proteins. And if we look at nucleic acids, so that's only comprised of 4 different nucleotides. So adenine, guanine, thymine, and cytosine. That's all that's found in DNA. So in large part, people debated the fact, could something made up of only 4 different nucleotides be responsible for all of the traits that we find in people? Additionally, different cells would have different proteins, and so it was largely believed that proteins were what were responsible for the genetic material. And that held true until about the 1940s. And in the 1940s, a series of experiments started to set out to prove what is responsible for that heritable material. So just to kind of recap, if we look in 1868, Friedrich Miescher was the first to isolate what he called nuclein. Today we know that as DNA. So he was the first person to isolate this. In 1910, we have Phoebus Levine, who discovers that DNA is only comprised of roughly 4 nucleotide bases. And he proposes what he calls a tetranucleotide hypothesis, in which nucleotides repeat over and over in equal proportions. This is later found to be not true. In 1928, a gentleman by the name of Frederick Griffith discovers that bacteria are able to transfer information from one to another. We call this principle, we learned about it last time, transformation. And that transforming harmless bacteria into deadly bacteria, that there must be a conference of heritable material. So it's this experiment that really sets us down this path of trying to answer what is responsible for our heritable material. And essentially what Frederick did, so Griffith took 2 colonies of bacteria. Okay, so this is Streptococcus pneumoniae, and what we find is in some colonies they're smooth. So under a microscope they look kind of smoothish. These ones are virulent. So if you put it into a mouse, it's going to get infected. It's going to end up dying. Whereas the rough colonies, if you put those into a mouse, the mouse will be absolutely fine. And what Frederick also found was that if you took smooth colony cells and you heat killed them, so you put them under extreme heat and injected those into a mouse, the mouse would also live. So heat killing the cells obviously kills the bacteria and makes it non-virulent. But the transforming principle that he discovered was that if he took those heat-killed smooth cells and he put it into a live culture of rough cells, something that normally doesn't kill mice, and he allowed those bacteria to grow, what he would find was that the mouse would then die. And when they analyzed the tissue of the mice, they would actually find living, smooth bacterial colonies in the cell. So he basically experimentally showed that something within these smooth-killed cells, even after they're dead, is able to be transferred into rough, which transforms them into a smooth colony bacteria. Now, in 1944, three molecular biologists by the name of Oswald Avery, Colin MacLeod, and McClin McCarty set out to decide once and for all, was there evidence that proteins were what was causing this transformation principle, or was it DNA or nuclein that was also isolated out of the nucleus? And the way they did that is they did a very elegant experiment in which they did an exclusion principle, where they slowly excluded every possible outcome to see what would happen using the same colonies that Griffith used. So they would take smooth cells, they would centrifuge those cells down to the bottom, they would heat kill those cells, homogenize them, which means they would basically crush the cells so everything is crushed, and they would extract the carbohydrates, the lipids, and the proteins from those cells. And then the first thing they would do is they would then put this into a colony of rough cells, and the transformation would occur, which would result in smooth cells being found in the control culture. So if you injected this into a mouse, it would kill it. Okay, the next step is they would treat this with protease, which would remove out all of the proteins. Okay, so now proteins have been removed out. They would then filtrate that, put it with the rough cells. So you have the treated, heat-killed, extracted smooth cells minus proteins put in here, and they would then transform it. And at this point, we would still get smooth cells. A transformation would still occur in these rough cells. So this led them to the conclusion that the active factor is not a protein. So proteins are not what is causing the rough cells to become transformed. They then treated it with ribonuclease. This would get rid of all RNA molecules. They would heat treat that, it was heat treated, it was then put with the rough colony cells, and a transformation would still occur. They'd still get smooth cells that would develop. So that says, okay, the active factor can't be RNA. Lastly, they would treat it with deoxyribonuclease, which basically shears up DNA. They would do the transformation, but this time they would not get any rough or any smooth colony cells, only rough cells. So in this very elegant assay, this very elegant experiment, they excluded proteins, RNA, and finally DNA. And it's only once DNA is removed out of this filtrate that there is no transformation. Now, you could say there's still carbohydrates and lipids in this experiment, but they knew that carbohydrates and lipids aren't found in chromosomes, so there was no reason for them to suspect those things. So at this point, they say DNA is accepted as the transforming principle. DNA is what's responsible for this. Now, even with this evidence, there's still a lot of people that said it just doesn't make sense. How can DNA do this? There might be some evidence, but we still think it's proteins. So there was additional experiments. We call these replications, in which they also tried to kind of look and can they prove what's going on. So in 1952, we have Alfred Chase and Martha Hershey, who worked with bacteriophage, so these viruses, and they did a very similar experiment in which they injected the bacteria, they labeled the phage, in which they grew that phage in a medium that had radioactive sulfur in there and phosphorus. Okay, sulfur is incorporated into proteins, phosphorus gets incorporated into DNA. So if they're in a medium that has a radioactive isotope, and then it gets brought in, that material would then be packaged into the DNA or into those viruses, and if those viruses infected, they could actually look and see what got injected in to the virus. So here we go. They grow up the phage in the radioactive medium. So these phage were grown in radioactive phosphorus. These were grown in a radioactive sulfur isotope, right? The phage then become labeled. So in this case, the DNA is labeled with radioactive isotopic phosphorus. Here the proteins are labeled with radioactive sulfur isotope. And then they put them into bacteria colonies, and they would then look at what did they get. And so what they found, first of all, if they take basically a X-ray of these cells, they would find in the ones that were labeled with the sulfur, they could see the radioactive viruses on the outside, but nothing inside that cell had radioactive isotope, meaning no proteins get injected into the cell itself. However, in the DNA, they could see it. And then when they looked at the ghosts, so those are the shells of the phage after it's injected, the ghosts were unlabeled. They were all unlabeled, whereas the ghosts of all of the sulfur ones were still labeled. And infected bacteria, they were labeled with P phosphorus. They would have viable P32 labeled phages that would get produced, whereas the ones with the sulfur, they were unlabeled, and any phages produced from these were also unlabeled. So this, again, gave evidence that DNA is what's being transferred and is what is responsible for the genetic material. So that is the vessel for inheritance. Now, a couple fun facts about DNA. First, all living organisms use DNA for information storage and a means of heredity. It's our blueprint. It contains the genes that make up all of our body parts. It is incredibly durable. So DNA can survive in ice. It can even survive in fossils, okay, for thousands of years. Even though we think of DNA, it's inside each and every one of our cells, it is a macromolecule. You can, in fact, see DNA with the naked eye. And to kind of explain that, how that's possible, in each one of your cells, if you were to take all of your chromosomes and you were to stretch out that DNA and lay it end to end, each cell contains 6 feet of DNA. Now, of course, it's very, 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 very thin, and it coils up on itself. That's why it can fit inside a cell. But that's, the length that we're talking about. Now, if we take every cell in your body and we did that same process of taking the chromosomes, laying them out end to end and stretching out, your body's DNA would be long enough to stretch from the sun and back 100 times. So that's an incredible amount of DNA that is contained within our bodies. Now, if we wanted to devise a genetic material and we want to have a molecule that's within a cell that can pass information one cell to the next, there's pretty much 4 characteristics that we would want that molecule to have. The first is that it's going to need to replicate. We would want it to be an adequate storage vessel, so durable. There needs to be some variable expression, so we don't want to express everything. We want to be able to turn things on and off. And then we would also hope that there's ways that we can gain variation so that things can change. If you look at replication, it's a fundamental property of all living organisms, right? So that information, in order for it to be passed on, it needs to be copied. So we double it, we partition it among daughter cells in mitosis, and in gametic cells, we half that information, form gametes via meiosis, and then we combine 2 gametes together to create a new organism. As far as storage, we talked about how durable DNA is, right? It can last even in fossils. It stores that genetic information even if it's not necessarily being expressed in a cell. And we have examples of how you can take organisms like this is a tardigrade, sometimes called water bears, right? You can put these things into the vacuum of space. They'll create these little cysts in which they basically become energetically like non-living. Their metabolism can't be measured when they're in these cysts. Yet when you restore them back, if you take them out of space, bring them back to Earth, they go right on replicating again. So, that just shows the durability of DNA in and of itself to continue to function. Expression. So there needs to be a way in which we can take that information and from DNA and turn it into RNA. And this gets us to our central dogma of molecular biology, how we go from DNA to RNA to proteins through transcription and translation. And lastly, the importance of having variation, that we can acquire new variations. So DNA has this ability. There is the possibility of mutation, so we can have genetic change, and that makes it responsible for the phenotypic variation we see amongst individuals and within a population. This occurs usually during replication, transcription, or translation that we can express this variability. So what makes up DNA? Well, DNA is made up of nucleotides. These are nitrogen-rich bases that are composed of each nucleotide has a nitrogen base. It'll have a pentose sugar. That means it's a 5-carbon sugar. If it's RNA, that sugar is ribose. If it's DNA, it's deoxyribose. So all nucleotides contain nitrogen-rich base, a pentose sugar, and they contain a phosphate group. And our DNA is comprised of thousands of paired nucleotides. Now, our two types of bases, purines and pyrimidines. Our purines, our nucleotides, are adenine and guanine. They have a 9-member ring. I'll show you that in just a minute. Our pyrimidines, they have a 6-member ring, and those are comprised of thymine and cytosine. Now, a way to remember these, pyrimidines, as you can see right here, have a Y in them, right? Pyrimidines, and both of our pyrimidines also have a Y in their name, thymine and our cytosine. Okay, purines, you can remember, have a 9-member ring, and both of our pyrimidines have 9 in the name, guanine and adenine. Okay, so I just kind of outlined those here, but here we can see, here are our adenine, guanine, thymine, and cytosine. So we can see the difference between our pyrimidines. They have that 6-member ring. The purines have the 9-member ring. Now you are responsible for being able to distinguish between these purines and pyrimidines if I gave you the chemical structure. And it's not that difficult. First off, remember the Y's, right? So our thymine and our cytosine have 6. So if we see a ring that has 9, we know it's going to be either adenine or guanine. Then we can start going to the number designation that corresponds with our nitrogen. Okay, that's going to be 9 for our adenine and guanine, right? 1 for our pyrimidines. The position of the NH2 then becomes important for distinguishing. So you'll see in both cytosine and adenine, guanine, we have these NH2s. We don't have an NH2 in thymine. So if it's lacking an NH2, it's a 6-member ring, we know it's thymine. Another way I like to do it, I don't have this written out here, but I also like to remember the oxygens. So cytosine has 1 double-bounded oxygen. Okay, thymine has 2 double-bounded oxygens. And you can remember 1 comes before 2, C comes before T. Okay, if we come over on the purine side, so adenine has 0 double-bounded oxygens, guanine has 1. So again, A comes before G. So whatever tricks help you remember it, just make sure you can identify them. Okay, so let's go through. Let's try these. First off, which ones are our pyrimidines? Which ones are our purines? Okay, remember the pyrimidines are only going to have 6-member rings, so A, and in this case D, are going to be our 2 pyrimidines. That would make B and C with the 9-member rings our purines. Okay, so then we can start looking at our NH2s. The one that doesn't have the NH2 we know is going to be thymine. Okay, the other pyrimidine is going to be our cytosine. So now all we have to do is designate what is the adenine and what is the guanine. And again, I can go off the ones that have our oxygen bound to it. So this one I see has an oxygen. This one doesn't have one. So that makes this adenine. And this one will be guanine. Okay, so make sure that you guys can go through that you can identify those. Okay, so we've talked about our overall bases. So again, here, this is the pentose sugar nucleotide base that we're going to have, right? Then we have our nucleotide, our nitrogen base. We then have our sugar. The difference between deoxyribose and ribose is going to be found on that second carbon. So if it's lacking in oxygen here, like we see right here, this would be ribose. If we're lacking that oxygen, we are deoxyribose. So that nitrogen base will attach onto our C1 carbon, and the phosphate group will attach onto our 5'. So when we put these together, we cleave off these OHs, and we call this is a dehydration reaction because we get a water is being removed from it. And so that phosphate always binds onto the C5. And the nitrogen base always binds onto the C1. And now we have our mononucleotide. So in this case, what mononucleotide is this? Well, this is a cytosine. And so this would be deoxy, because again, we don't have an oxygen right here. So it's deoxycytolilic acid. That's the mononucleotide. If it was an adenine, it'd be deoxyadenylilic. If it was a guanine, it'd be deoxyguanylilic. And if it was a thymine, deoxythymidylilic. Okay, if it wasn't DNA, if this was ribose, then it would just be adenylylic acid, guanylylic acid, thymidylilic acid, cytidyllic acid. So let's try to assemble, okay, our deoxyadenylylic acid. Where is our H2O going to be removed? Well, we're going to move out this guy. We're going to move out this one. We're going to move out this and this hydrogen. All right, and there we go. That's how it would look. So, our DNA is comprised as we have this process continues with thousands of linked nucleotides. We call that a polynucleotide strand. And we number those strands based off our C5 and our C3 sugars, because this is where the next nucleotide base is going to attach, is right here. Right? And on our 5'. So we'll have the 5' end will attach here, and we do that through what we call a phosphodiester bond. Another aspect of our DNA is we have 2 strands. So we end with 1 second polynucleotide, and then those 2 strands, they get oriented what we call anti-parallel to each other. And that creates this ladder. And the reason why it has to be anti-parallel, if it was parallel, we would not be able to have our hydrogen bonding between our nucleotide bases. That's what creates the rungs, or of the ladder, the base pairs. Okay, the polynucleotide strands, those sugars and phosphates, they contain the sides of our ladder. all bases have specific complements. So adenine, we always have a purine to pyrimidine complement, and adenine will always bind to thymine. Guanine will always bind to cytosine. Okay, in DNA, and this brings us to Chargaff's rule. So I told you about Levine's tetranucleotide hypothesis, in which he said all of these are found in equal proportions in DNA. That's not true, because adenine will only bind to thymine, guanine will only bind to cytosine. So what that means is you have an equal number of adenine to thymine, and you have an equal number of guanine to cytosine. So I could ask you a question on a test saying, hey, if 20% of your nucleotides are adenine, what percent are cytosine? Well, if 20% are adenine, that means 20% are also thymine. So that equals a total of 40% being ATs, which leaves 60% being GCs. Divide that by 2, that means 30% of my nucleotide bases in that DNA strand will be cytosine. So let's look at these anti-parallel strands. So again, here we're just seeing, two sets of mononucleotides being bound, but here's our 5' end, so the 5' carbon. We're going towards a 3' carbon end on the base. So there's our deoxyribose. Here is our phosphate, and then the base is oriented towards the interior of the DNA strand. And you'll notice again, guanine pairs with cytosine, and in this anti-parallel structure, because here's the 5' over here and the 3', this allows the hydrogen bonding to occur. If we didn't have this, if they were in parallel, we would not be able to have this paired hydrogen bonding. They would basically be facing in the wrong direction. The hydrogen, the bases would be facing over here, and we couldn't do that. Between a guanine and cytosine, we have 3 hydrogen bonds. Okay, between a thymine and adenine, we have 2 hydrogen bonds. That means it takes less energy to break this bond than it does that. Again, if we were joining these together, that's where we'd have our dehydration. Our phosphodiester bonds would get made, and we would have this. So our strands, because they're an antiparallel, they ensure the dimensions of the molecule are even due to base complements, and that we can have that hydrogen bonding. We get these 2 strands, and what this causes is that the molecule begins to twist around each other. So it twists in a clockwise direction. We have 1 full twist for every 10 base pairs that we find. It also ensures, because of all of the sugars and phosphates being on the backbone, those are hydrophilic, meaning they like water. So water is attracted to this. But in their base pairs, those are hydrophobic. They don't like water. So water doesn't enter into the interior. We create this protective shell of water around the DNA. This is part of what keeps it so stable and durable in our environment, is the interior is basically being protected by the shell of water. You can follow this website if you want. This will actually bring up a virtual DNA molecule, or you can look at the twist, you can pull things apart, and kind of see how things pair up. Last thing that this chapter talks about is the different types of DNA we find in our cells. So we have 3 varieties of DNA. We have our nuclear DNA. This is what we think about when we see chromosomes, right? It's found in the nucleus, contains all of our chromosomes that are passed on from our mothers and our fathers. We also have mitochondrial DNA. This is found in mitochondria, and we have chloroplast DNA, found inside the chloroplast organelles of cells. So nuclear DNA is in the nucleus. It's linear. It carries all of our phenotypes. In the human genome, we have at least 30,000 genes. Okay, and again, 50% is going to be inherited from the mother. 50% is inherited from the dad. Mitochondrial DNA. This is circular. Okay, it's similar to prokaryotic DNA. In a bacteria, we have that plasmid. It's circular. It only contains 37 genes. These 37 genes are the genes that control cellular metabolism. So the processes of the Krebs cycle, electron transport chain. And it's only inherited from the mom. Why from the mom? Because when we have a sperm fertilize an egg, it injects its nuclear DNA in, but it doesn't inject any mitochondria into that cell. So that means as the mother's egg cell divides, the mitochondria are what are getting copied and pass down from cell to cell. And all those mitochondria contain the maternal mitochondrial DNA that the mom had. Chloroplast DNA, very similar to mitochondrial DNA. It's also circular. There's more genes. There's about 120 genes, and these are the genes responsible for photosynthesis in plants. And this is a little bit different than say mitochondrial DNA. It can be inherited both from the mother and the father. The very last part. So this is a diagram. This is what we'll be talking about in this unit. Going forward is what we call the central dogma of molecular biology, how we go from DNA to RNA by the process of transcription, how we replicate our DNA. We'll talk about that next time. So DNA replication. Transcription turns it into either messenger RNA, ribosomal RNA, or transfer RNA. Then we move those to the ribosome, our mRNA and tRNAs, and we then translate that into a protein.